Thank you, thank you, thank you. Handing it over to Peggy Wallace.
Yes, welcome to the gene therapy session. I'm Peggy Wallace, an NF1 researcher at the University of Florida, and I've been helping with the CTF's NF1 gene therapy initiative. And I'm going to let my two co-panelists introduce themselves quickly. Kat?
Yeah, hi everyone, Kat Meyer. I'm a principal investigator at Nationwide Children's Hospital in the Center for Gene Therapy. I'm working on gene therapy for neurological, neurodegenerative, and neuromuscular diseases. We have several programs that we brought into the clinic so far. Very, very nice to be here.
Mike?
Morning all. I'm Mike Wooten. I'm the CEO of NF2 Therapeutics. Previously, I founded Inflection Therapeutics along with Dr. Scott Plotkin and Mark D'Souza, where we developed the first topical MEK inhibitor for cutaneous neurofibromas in NF1. The three of us have also co-founded NF2 Therapeutics, and the two key programs within NF2 are a gene therapy program for NF2 and an antibody drug conjugate targeting meningiomas in NF2.
All right, so today the three of us are going to present considerations in gene therapy research. And to stay on time, we're going to hold questions until the end of our presentation. Ideally, put your questions in the chat box. And if we have time, we can go live to some of those as well. So I'm going to start with a general overview of gene therapy. By definition, it's the use of genetic material to treat or prevent disease. And that doesn't have to be necessarily hereditary disease. There's a wide variety of choices, and the choice for strategies is based on addressing the underlying problem, which has to be well defined at the biological and molecular level. There are strategies that span from efforts to replace, to knock down, to add in, to edit DNA and RNA, lots of options for each part of the overall strategy. And so there's multiple factors that have to be considered in designing gene therapy. I'm going to talk about them in general, and then my colleagues are going to talk about them more specifically related to NF2. So one consideration is the choice of your target cell or target tissue that is going to receive the therapy. It's usually the cell that has the abnormality that's causing the condition or symptom, but it's possible in some disorders that the therapy would be a secreted protein, in which case other tissues could suffice as a source of the expression. You also want to have consideration about the choice of the intracellular target that is going to be within the nucleus, such as a gene or transcript, or have a therapy that functions from the cytoplasm, such as from an episome. There's also decisions to be made about whether to attempt a permanent, potentially one-time only treatment versus developing a temporary therapy that is going to involve subsequent treatments. And again, these are options that are out there, and these you have to consider your disorder and the actual underlying molecular issues to make these decisions. Choosing a vector, which vector to use to actually deliver the gene therapy. That can be a viral, such as adeno-associated virus, one of the more common ones, or a non-viral mechanism, such as use of nanoparticles. You also need to consider whether it's going to be a systemic delivery or a limited in vivo delivery. And with some disorders, there's an option for ex vivo gene therapy as well, for example, which is being done now with sickle cell anemia. Researchers also need to determine what is the level of efficacy and efficiency that I need to produce a therapeutic effect. That's part of the research that goes into testing the strategies. And probably of the top importance is safety. So there are many control and safety factors that are involved in deciding which strategy you're going to use, and also going through the research and heading it into clinical trials. And finally, you also have to think about feasibility. A vector might have a very effective result, but if you don't have enough of the actual vector, for example, you have a difficulty manufacturing high yields, then it's not going to necessarily be something that will be feasible to produce because it'll be prohibitively expensive to take to the clinic. So you can see there's a wide variety of kinds of issues in gene therapy. A lot has to go into the design. And related to NF2, ideally, the great thing to do would be to prevent NF2, Schwannomatosis. but that's much, much harder to prove in a clinical trial type of setting than shrinking an existing tumor. So based on this feasibility, the gene therapy efforts are predominantly focusing on treating existing tumors. And Kat is now going to talk about these considerations.
Yeah, thank you, Peggy. Yeah, so for me, you know, when I think about NF2 in terms of gene therapy, it's a little bit different from what we're usually doing here, because for the gene replacements that we have in clinic so far, they're classic loss of function mutations, and you know, you're basically trying your best bet to target as many cells as you can. And the more cells you target, most likely will have a better outcome, because you are just basically have more healthy cells that will have a better function. And technically that might be true for NF2 as well, but every single cell you miss, and that's the difference here, actually can still become a tumor. So the dosing is going to be a much bigger issue, or a much bigger question mark here, I would say, when we talk about prevention of disease. There is really no way at the moment to basically deliver gene therapy to every single cell that is in need. If you do that, you will probably have to dose extremely high, and a lot of the dosing systems that we have right now, they create hotspots, so you will overdose certain areas at the same time. So that's a really, really big question here. You know, how do you achieve sufficient targeting, and what is the sufficient targeting for NF2 so that you can prevent tumor growth? But on the other hand, you know, if you can't prevent 100% tumor growth, it might still be worthwhile. You know, if you could reduce the tumor burden, the future tumor burden of the patients by 20 to 30%, I think it would still probably be worthwhile doing that. So in the end, it might still be the same situation. The other problem that we're having, I guess, with the prevention strategy is, of course, you would want to treat the patients before they have tumors, or, you know, but you can't really predict how fast the tumor growth is going to be, or in many patients, you might not be able to predict that if they are diagnosed very early on. And so that makes the clinical trials very difficult, because it's really, really hard to find funding for a 10-year clinical trial, which is what you kind of would potentially need, or for a very large clinical trial in order to really show that you are reducing the number of tumors or how fast they grow. So those are very difficult tasks, I think, for the classic gene replacements. And then another aspect is also, you know, how long would the AAV expression last? So nobody really knows that. That's a problem for all gene therapies. I mean, we're in the clinical trials right now about, you know, 5 to 7 years post-dosing for those more systemic targeting. So far, so good. But nobody knows how long these treatments are going to last. And in particular, if the cells are dividing, because AAV does not, or to a much lesser extent, integrate into the genome, you might be losing, you know, efficacy over time. And that, again, if you're thinking of that 10-year trial, if you're losing efficacy over time, the tumors might start growing, or might start growing faster, which will complicate the analysis. So all of that, I think, is something that is pretty unique, not necessarily just for NF2, but in general for NF gene therapy. And then, of course, as an alternative, we can target the tumors directly, as Peggy said, but then you lose that advantage from the one-and-done kind of treatment. So you'll have to go in and target the tumors multiple times, and you probably know much better than I do. You'll have to potentially do some complicated surgeries to access the tumors. And, you know, the other question there is, again, the same one as with the general treatment. How many cells in the tumor do you actually have to target to make sure that tumor doesn't continue growing? Now, I think, again, from a patient perspective, even if we can slow down tumor growth instead of completely stopping, that would already be a big success in many cases. So it's definitely still worth doing. But I think one thing that's really important from my point of view is to manage the expectations, because sometimes in the patient communities, expectations are really, really high for gene therapy. And you know, some people talk about a cure for these diseases. And that's generally, in my opinion, not true for any disease, but especially here, you know, with all the challenges that we have to work through, it could be potentially a very good treatment, and maybe the prevention and the tumor treatment directly can be combined, but it's not going to be a cure. The patient still will have cells that have the potential to develop tumors, and so the combination treatments, I guess, moving forward will be really, really important. So I think those are the challenges, and challenges are always good. We just need to address those. But I think there is a lot of things to consider for these gene therapy trials. And Mike, you're going to talk a little bit more details about additional challenges on how to develop these, right?
Absolutely. So I'm going to quickly go through some of the key questions and challenges faced when thinking about developing a gene therapy for NF2. And one of the first of those is target. What is your target? Are you going to go after Merlin directly? Or are you going to choose to go after a related pathway, perhaps associated with proliferation or contact inhibition? Are you going to target tumor growth? Are you going to target targeted cell killing? And then another key question is, what is it that you're targeting? Are you targeting schwannomas, meningiomas, ependymomas, peripheral tumors? And that raises the issue of specificity. One of the challenges with respect to AAV-based gene therapy is the key drivers of specificity are the capsid serotype and the tropism of that serotype towards certain cell types, as well as the promoter upstream of your gene and the specificity of that promoter towards certain cell types. And so those two in combination can give you a highly specific targeting capability. But backing up, if your intention is to target Schwann cells, arachnoid cells, ependymal cells, all at the same time, what are the odds that that same combination of capsid serotype and promoter are going to be exquisitely specific for those cell types and avoid off-target delivery? So that's a key challenge. Physically, how are you going to deliver it? Is it going to be IV, intrathecal? Is it going to be intratumoral? Is it some other method of delivery? And then from a safety perspective, dosing has already been touched on in AAV-based gene therapy with extremely high dose levels. There have been fatalities. What will the overexpression of Merlin be? What will the result of that be? And what other off-target effects could there be? There are also a number of toxicities which are on the radar of FDA. In September last year, the AdCom met specifically to talk about AAV-related toxicities, liver toxicity, DRG toxicity, thrombotic microangiopathy, brain MRI abnormalities, post-intraparenchymal administration are all things that are on their radar. The good news is that none of those are really viewed as showstoppers. However, they will certainly add to the preclinical studies, the work, the amount of data that's required to take any therapy into the clinic. The next set of questions are around efficacy, and we've talked about what percentage of cells need to be transduced for prevention or to drive regression. What is the number of copies per cell? What is the rate of expression that you're targeting to drive progression or prevention? And then in terms of demonstrating that in animal models, if you're looking to demonstrate regression, de facto, you're going to be working in older animals with established tumors. If you're looking to demonstrate prevention, you're going to be working in younger animals, and depending on the stage of development, what about cell division? as was just stated, cell division will rapidly inactivate a gene therapy, which is both a factor in terms of demonstration in animal models, but also what the ultimate design of your therapy is going to be. Another question is, what about endogenous mutant Merlin? What impact will that have? If you're restoring a full-length trans protein, what will the impact of the endogenous mutant protein be? Neutralizing antibodies are another challenge, and you've got to think about not only previous exposure to the virus that you're using as your capsid, but also cross-reactivity between exposure to other AAVs that may be cross-reactive with the serotype that you're using. And then questions about animal models. So do cellular models translate into the animal experience? Which animal models best recapitulate the human condition? Also questions about, are mouse models effective systems for evaluating AAV-based gene therapy? And certainly from FDA's perspective, they expect non-human primate studies for safety and toxicity for AAV-based gene therapy to move forward into the clinic. Some of the commonly used animal models, Marco Giovannini's mouse model, Wade Clapp's mouse model, Longsheng xenograft meningioma, and schwannoma are all models that are commonly used in gene therapy, and obviously other therapeutic development efforts as well. Next, Kat Meyer is going to take us through some of the gene therapy development efforts that are currently ongoing.
Thank you, Mike. Yeah, so ultimately, you can already imagine from what we said, there's a couple of approaches that people have been working on. The first one is obviously a classic gene replacement, where you would attempt to re-express some form of the NF2 protein, Merlin. Can be modified, phosphorylation resistant versus non-resistant, could be driven by its own promoter or truncated versions thereof, or by other promoters that can be selected. And there are several groups that have been working on this. Another approach is what we call reduce and replace. So you would actually attempt to downregulate the endogenous NF2 protein in the same cassette where you're going to re-express a new form, a healthy form of NF2. And this will be really attractive because in some cases we don't know if the mutated proteins still have additional function or a gain of function, as well as whether you could get overexpression of the endogene of the NF2. So the reduce and replace will kind of address those questions as well, but you have to be very careful to make sure that your modified transgene doesn't induce an immune response. Sometimes when you modify the sequences, they can be recognized as foreign, especially if they're very GC rich. Another attempt that is followed is a suicide gene delivery. So actually deliver genes to the tumor that will kill the cells or will stop the growth of the cells. Another one that's currently explored is an antibody against VEGF, which seems to be upregulated in vestibular schwannomas. So the AAV can actually encode for an antibody or a mini-body that can target such pathways. Unfortunately, that can be toxic if it's done systemically. So potentially would have to do that more locally as well. And then, of course, everybody talks about gene editing. So you can actually go in and change that gene itself using especially CRISPR-based approaches. However, unfortunately, at the moment, those are still relatively inefficient. So you're not going to get your genome changed in every single cell. So that's the same problem there. Also, there are immune reactions that have been observed against the Cas9 protein or other Cas proteins. Since it is a bacterial protein, if you do that intratumorally, it might not be a problem because maybe the immune system can help clear those tumors, but it's just something to keep in mind as well. All of the...
Pat, I think we're going to go ahead since we're running out of time, almost time for the break. You're almost done. But I think what we'll do is let's take just a couple of minutes to open it up for questions. Since we did start a couple minutes late, does anybody have questions right now? Or you can send them to chat in the meantime. I don't see any questions at the moment. Okay. So if you think of any over the break, go ahead and send them. And that's the wrap up of the complicated area of gene therapy and NF2. All right. I guess it's time for break now, right? Wait, what about tomorrow? Okay, we'll check on this in just a moment.
Do we really need non-human primates? I think it depends on the clinical trial design. If you just do intratumoral injections, you might get away without the NHPs. For more systemic applications, you most probably, almost definitely will need NHPs, depending especially also on the serotype you use. Mike, do you want to add anything here?
You know, at this stage, my experience has been that FDA really wants that data, and it's driven by the belief that the AAV in reaction to AAV is most closely recapitulated in NHPs versus other animal systems.
And it might be different if it's a non-viral delivery. Matt had a question about prime editing, which is part of base editing. It's always one of the options. But we have to, again, very carefully think about, is this going to be one of the better approaches for as many NF2 patients as possible? So it's certainly one of the things on the table. Somebody else asked about prime editing. You want to say anything about that, Mike, real quick?
Yeah, I don't think I have anything to add.
Right.
Yeah, I think, it's the same, I would say has the same drawbacks. Efficiency could have off-target effects, and then it still doesn't solve the problem of how many cells do you have to target. I mean, I think that is, and sorry, maybe we've been showing a lot of the challenges, but I think it's important to raise those so that we can think about them ahead of time. Doesn't mean that it's not going to work.
Reality, right?
Yeah.
Thank you so much, Becky, and the whole panel.